Okay. Hello? Hör du mig? Okay, most welcome everybody. My name is Stefan Ståhl and I'm the chairman of this part of EVA relating to biotechnology and life science. And I'm extremely happy to see you all here. This is, we couldn't be much more than this. And I will steal minimum of time for introduction. I will more like to say that we will be very stringent that you use the microphone asking questions because this is also streamed and so on. So we have to wait for Maria's running feet to give us the mic. And I'm extremely honored and happy to have Fredrik Bäckhed here and the team. And he will tell us about the relation between the gut and its microflora and how we feel. I think that has something with gut feeling to do so. He's from the West Coast. So, the floor is yours.
Okay, thank you very much. It's really nice to see everybody here today. So we will spend the next 2 hours or so discussing the microbiota. And we will do that from 3 different perspectives, but the common theme is that we will focus on its role in metabolism and its how we can analyze it, and potentially then finish off with our foreign guest, Max Nudorp, who will go on and tell us about the treatment potential. So the 3 speakers today will be me. I'm newly elected to EVA, and I have my basis in Gothenburg. Then it's Jens Nielsen, who's got the gold medal from EVA a couple of weeks ago, who's also at Chalmers University. And then Max Nudorp from Amsterdam. We will talk about 30 minutes each, and then there will be 5 minutes question after each talk. And then we will finish up with a 10-15 minute general discussion if there's questions that you have come up with in the first talk and forgot about and want to then ask them at that point in time. So, walk over here. So, what are the things we will talk about today? These are the main players of this seminar. These are the bacteria that we have in our gut. They are closely associated with the colonic epithelium. So these are the human cells we have. These are the bacteria. The human cells produce mucus that the bacteria can be embedded in, and which also protect us from too much of a direct contact with the bacteria. And then we have the food particles. So what we eat, the bacteria will see, they will digest, and they will, as I will show you in a minute, form bioactive compounds that can then affect our physiology. But what are the microbiota? So these are the microbiota. When I say the word microbiota, it's the same as Tarmfloran in Swedish. It's all the bacteria we have in this environment. And in the gut, it's estimated we have at least as many bacteria as human cells, but many others says that we have up to 10 times as many bacteria as we have human cells in the body. So every one of us that in this room have, we are about 10% human and 90% bacteria. We also like to view the bacteria or the gut microbiota as an organ. So it's sort of the forgotten organ in the body. We talk about the liver, we talk about the spleen, but we also have the gut microbes. And this organ weighs about 1 kilogram, and it's formed by 500 to 1,000 different cell types. And then each bacteria has its own genome, just like we have our own genes. And if you tally up the genes the bacteria has, they outnumber our genes with almost 1,000-fold. So on a genetic basis, we're less than 1% human and 99.9% bacteria. This is just to stress that it's not only in the gut we have bacteria. On each surface of the body, we have bacteria. So on the skin, we have 7 different kinds of microbiota. We have a different bacteria community on the nose, which is a little bit colder. And then in the armpit, that's a little bit more moist and warm. But of course, we then have different bacteria also in the stomach, in the vagina. But what's really amazing, I think, if we look at the microbial composition on a more global level, is that in the gut, in the colon, even if we have up to 1,000 different species, they belong to just 2 main groups of bacteria. And the reason for this is that this is a very a consistent environment, about 37 degrees, with a lot of nutrients coming in there. So we have specialists, and these tend to belong to these two groups of bacteria. If you then instead start to look at the skin, we have more colors. It's a more diverse habitat, although we have a generally consistent temperature. The temperature can fluctuate. We're exposed to different environments. So you have more of different kinds of bacteria that are adapted to different life situations. Similar, if we would go out and take a gram of soil from the ground, we would maybe have 100 different of these groups of bacteria. Because that environment fluctuates much more, we can go from -10 to +30. It can rain, it can be sunny. So you have more bacteria that can sort of flourish at different occasions. Another point I'd like to make is that just because a scientific field is hot, it doesn't mean it's new. So right now, the microbiota is a very hot topic, but people have been studying it since the 1600s. This is Antonia von Leeuwenhoek. It's nice to be at this Academy of Engineering, since this is really a pioneer. He developed the microscope, the first microscope. And when he founded the microscope, he wanted to use it to something important. And one of those important things was to look, what does he have in his mouth and what does he have in his feces? And he could describe that there were different kinds of organisms. He didn't know what they were. They were too small to be an animal or cell. And they still were living, so they were not molecules. So we call them animalcules. And these were then the bacteria, and he could describe that he had different kinds of bacteria in the gut. And these were either could be small, they could be motile, they could be large. And when he had an episode of diarrhea, their composition changed, indicating that you really can affect your microbiome in your disease, and we call that generally dysbiosis. Today we have a little bit more of elaborate means to look at the microbiota, and I just will have one technical slide. So what we do now is that instead of looking at the microscope, or what we have done for a long time is to culture bacteria on different plates and then look what they are. One of the problems with this methodology is that it's been estimated that we maybe can grow or culture about 10 to 30% of the bacteria in the gut, because the gut is anaerobic, there's no oxygen. And if you culture something outside, there's oxygen, and many things are hard to culture. They can also have very specific growth requirements. So it's neat in the way that we have the bacteria, but it's a very, very low throughput. You need many students to culture each bacteria, basically. However, I think we also have overestimated the difficulties. It's not impossible. It's just very, very hard to culture them. So what we do instead is that we use the genetics of the bacteria, the genes of the bacteria. Either that we can look at one specific gene, which is called 16S rDNA. It's a component of the ribosome, the thing in the cell that makes all the proteins. And we can then get some kind of a map on which bacteria that are present. We can do that by several different means, but we can also choose to read the genes of the entire microbiota, and then we can look at what functions and what are the bacteria that are present there. So we look at it and we call that bioinformatics, and this is something Jens will talk more about in the next talk. So one of the things that really separates our genome from the microbiome, as we call the combined genes in the microbiota, is that our genes are relatively stable over time. The genes we were born with, we may almost die with. There's some mutations along the way, but generally very few changes in the genes. In contrast, our microbial friends can be shifted by many different means. Either if we eat antibiotics, we kill a lot of bacteria, and then we change the gene pool in the microbiota. But also other medications, and I will give you one example in a couple of slides, demonstrating that regular medication we take can affect the composition of the microbes. We know that if we put mice in cold, they change the microbes. And especially the diets we eat will have an effect on the microbiota composition, such as if we especially remove fiber from our diet or increase amount of fiber in the diet, we dramatically shift the microbiota. And we shift it in a way that the bacteria that can eat fiber will expand if we have fiber. And fiber has many different linkages. It's a series of sugar molecules, and bacteria are much better than we are in degrading the sugar. So if you eat a lot of fiber, you have more bacteria. You have an increased richness or diversity. But what's interesting as well is that the bacteria we have that are affected by the by the diet we have, they then digest the diet and they produce bioactive compounds. Like short-chain fatty acids is a very common molecule. You may have heard about butyrate, smörsyra in Swedish. That has many beneficial effects. So this is another point I want to make, is that not only the microbiota we could consider to be an organ, we can consider it to be an endocrine organ because it produces signaling molecules that then reaches other tissues in the body, such as the adipose tissue, the liver, the brain, the cardiovascular system, and even the lungs, where it can have effects on the gene transcription in these organs. So as you can imagine, since the microbes can have so many different effects on what we eat and change in health and disease, there's been a number of diseases and syndromes which has been associated with an altered microbiota. And one of the points here is to make is that most of these studies and most of these diseases, and I will go through some of them, is what we call associations. You compare a healthy group of subjects with a group with a disease, and you find a difference. And if you do those kind of experiments, you can find differences in everything from patients with stroke, but also subjects that have an extreme stress situation, or children with an autism spectrum diagnosis. We see differences in microbiota in patients with allergic asthma or with atopic dermatitis, but also in a whole series of metabolic diseases. And this is the thing I want you to remember when you open the newspaper in the morning and you read in the scientific part that maybe a disease X has been associated with an altered microbiota. It doesn't mean that the altered microbiota causes disease. This is where I think we really need to work towards now, is the cause and effect relationship here. So in my group, and this is why I had the pleasure to pick the topic of today's seminar. So I put on metabolism, and that is coming from a study we did when I was in the United States for 15 years ago. We wanted to address whether the microbes can affect the body composition. So can it affect how much fat we have in the body? So we compared something we call germ-free mice. These are mice that never seen a bacteria, with normally conventionally raised mice. These are regular mice. We put them in a machine that can determine fat distribution, and we find that the germ-free mice have significantly less fat than those that have a normal microbiota. What then was interesting is that we take these mice and we colonize them with a normal microbiota for 2 weeks. We can normalize the amount of fat. So these are the same mice as these mice, but they've had bacteria for 2 weeks, and then they've increased the amount of body fat with 40 to 50%. So in an animal situation, it's pretty clear that the microbiota contributes to how much fat we have in the body. But this is probably what causes symbiosis between us and the bacteria, was that the bacteria will help us to digest fiber when we don't have a lot of energy. So it's a good thing. These are probably, without bacteria, we're malnourished. But we also did the converse experiment where we took germ-free and colonized mice and we put them on a Western-style diet. These are diets that have energy from protein, sugar, and fat. And then we see that the germ-free mice do not develop what we call a diet-induced obesity, whereas in the presence of bacteria, they gain a lot of weight. So they have gained about 25% in the body weight compared to those mice that have no bacteria. I'm going to skip that, and then we move here. And the reason for that is obesity is interesting, but the problem we see in obesity is that in patients we see an altered microbiota in obese individuals. But in all the studies that have been done, it's not a common theme because they have been very small and there's no consensus really. The only consensus that's seen is that those individuals with obesity has a little bit reduced diversity. There are fewer bacterial species, but we think that's mainly due to that they eat differently. They eat less fiber, for example, maybe more fat, more sugar. And since it's been controversial what is the common theme, and we're not really working on that so much in my laboratory, I wanted to fast forward us into the field of type 2 diabetes. And together with Jens, who's speaking next here, we performed a study in Gothenburg where we looked at the microbiota in patients with type 2 diabetes. And at the same time, there's a group in China doing the same thing. And what we found in both of the studies as a common theme is that if you have type 2 diabetes, you have reduction of butyrate-producing bacteria. And we think this is very interesting, and this has been confirmed in other studies. So in contrast to the field of obesity, in the field of type 2 diabetes, we see a commonality that the type 2 diabetes subjects have reduced levels of this bacterium. But one thing that became evident is that we have to be careful, because there came another paper describing that maybe we're wrong, because if you take the diabetes medication metformin, which is very common in type 2 diabetes, you have the effect of the drug has an equal strong effect as the disease. So we have done an experiment where we wanted to address that in more detail. So together with José Manuel Fernández Real, who's in Spain, we took a very scientific approach, I think, where we took 40 individuals with type 2 diabetes, and we randomized them either to metformin or a placebo, so a sugar pill. And we then studied how the microbiota developed. And now I will show you some more of what we call data slides. So I want you to expose you to what the kind of data we see in the laboratory. And I will guide you through it, so it's not going to be too complicated, I hope. So I told you that we are assessing the microbiota by doing sequencing. And then we have see that we have different bacteria that can be altered, and we put them in different colors. So bacteria that are not changed, they are in these very weak colors. Bacteria that increases a lot are in dark red, and those that are reduced are in stronger blue colors. And then we have those patients that were on placebo before they started the treatment, 2 months after and 4 months after. And you can see really nothing changes. But in the patients that got metformin, you have some bacteria that are increased after metformin treatment and others are reduced. So that was really exciting. We saw differences. But does it mean anything? And then we can do an experiment where we transfer the microbes into germ-free mice to see if the microbiota can have an effect on the disease outcome. And when we do this, and this was work by Robert Cesar, who's in the group, we then take feces from a subject before and after metformin treatment, and we put them into a mouse, and we don't see any effect on body weight, we don't see an effect on insulin, but what we can classify subjects with diabetes with is that we can give them a glucose load. This is what a lot of pregnant women do during the pregnancy to see if they get a gestation diabetes mellitus. So basically you drink glucose and you see how you respond to that glucose. We do the same thing with the mice, and we see that mice that got their microbiota without any treatment, they had a much higher glucose levels than those that were colonized with bacteria after metformin treatment. So this is could show then that if we take metformin, the microbiota is altered, and that then if we transfer that to mice, you have an improved glucose metabolism. So we suggest then that perhaps one of the ways metformin works, which is a very common diabetes medication, it works by altering the microbes. So why do we do all of this work? Well, of course, we have to have a vision. And the vision we have is that the group of us in here, for example, in the future, we'll not take a blood sample when we go to the general practitioner. We will actually take a fecal sample and look at which microbes we have. And then maybe we can find some of us that are at risk of developing type 2 diabetes or other diseases for that matter, and then stratify the treatment into a more personalized treatment where some that are maybe lacking these butyrate-producing bacteria, they get a drink with butyrate-producing bacteria. Others, we can advise which kind of diet they should be on to maintain an improved metabolism. I think we all will benefit from exercise, but perhaps we can then talk about who should have which drug to treat that disease. I mentioned then metformin, but there's also a lot of new knowledge now that some of the modern cancer treatments, they need the microbes to be activated. And also that potentially that we can select those that would benefit the most from bariatric surgery. I will continue by showing a few slides on what can happen when we interaction between bacteria and our diet. And in collaboration with Inge Björk and Anne Nilsson, who's at Lund University, we wanted to address the role of microbes in fermenting dietary fibers. And the way we did this is that we selected 40 people, 40 humans, and we gave them a barley kernel bread. This is a healthy, so-called healthy bread that they've used a lot. And they said that this is, we really think that this will change the microbes. And I asked back then and said, well, do you see that everybody gets improvement, or do you see that some has an improvement and some do not? Well, she said, it's actually that not everybody improves. I said, okay, let's try that. So we took these 40 individuals, we fed them the bread for 3 days consecutively, then had 2 weeks without any bread, and then they got a white wheat bread as a control. And also in the humans, we do this kind of glucose tolerance test, as I mentioned, that we did with the mice. And what we see then is that in those that do not respond to the diet, the glucose excursion is similar, either if you have had the wheat bread or the barley kernel bread. But those that respond, they have an improvement in both the glucose and in the insulin signal. So after the barley kernel bread, they have the green dots, which have a better glucose excursion curve. And what was interesting is that we could then find that those that responded, they really had a bacteria called Prevotella. It was much more common in these subjects, and they really increased when you ate the bread. So 2 points here is that they had slightly higher levels already before they ate something, but then after they have eaten the bread, they really expanded the amount of this bacteria Prevotella. And Prevotella is known to degrade fiber. So to see whether it really matters which bacteria we have, we then colonize our germ-free mice again with either the microbiota from the responders, the humans that responded to the treatment, or those that did not respond to the treatment. We then look at the glucose tolerance test 2 weeks later, and what we find is that if we colonize the mice with non-responder feces, it doesn't matter what we give them, they have a similar glucose excursion curve. But if we instead colonize the mice with those microbiota that responded, from those that responded, we see that the mice that got the baseline microbiota without any barley kernel had worsened glucose tolerance compared to those that actually got the barley kernel bread microbiota. So I see I'm running out of time a little bit, so that's why I had to speed up. But I also wanted to share some of the data with you on bariatric surgery. And bariatric surgery is a quite common procedure. We do 8,000 bariatric surgeries per year, and this is weight reduction surgery. And what you can do is you can do it in many different ways. And we see that you can either do a Roux-en-Y gastric bypass, where you disconnect, you make the stomach very small, and you then bypass the stomach and the upper part of the small intestine and attach that further down in the small intestine. Or you can reduce the size of the stomach, which is called vertical banding gastroplasty. The reason why I like to show this data is that a lot of the knowledge we have is from Gothenburg, from Lars Sjöström, who's running this study since many years, where you have we've followed these different surgeries and how they function over 15 years. And if you don't do anything with obese subjects, they may remain obese. If you do a Roux-en-Y gastric bypass, you can reduce the weight by 30% almost in 15 years, or after 15 years. And vertical banded gastroplasty is somewhere in between. And as you can expect, that if you do these changes to the microbial, to the stomach environment, you also shift the microbes a lot. And what was furthermore interesting is that if we once again colonize the microbes, the feces of these patients into germ-free mice, we see that the obese, that the ones that receive the obese microbiota gain more weight than those that receive those after bariatric surgery, once again suggesting that the bariatric surgery can have its effect by altering the microbes. So the final couple of slides is something that I like a lot. We're going to go a little bit into what we call for mechanism, because what I tell you right now is that we see that after surgery, after drug treatment, we have an altered microbiota, and this can contribute to the treatment outcome. But how does it work? And I'll give you a snapshot of what we can do, and I will continue with this bariatric surgery story. And for you who paid attention for the first slide, one of the signaling molecules that the microbes are doing called bile acids. And just very briefly, bile acids are produced in the liver from something we know as cholesterol. You get bile acids. These are then modified by the bacteria into what we call secondary bile acids. And the important thing is that these then, I like to call hormones, because they're released by the liver, they're modified by the gut microbiota, and then they're out in the body where they can bind to different receptors. And what we find is that after bariatric surgery, we have a much increased release of these bile acids, especially these secondary bile acids that are microbially produced. And we can then show that they signal through a molecule called FXR. And what we like to know is to have, does this altered microbiota after bariatric surgery signal through FXR? We can measure a reporter called FGF19. And we do that, and we see this beautiful elevation telling us that Roux-en-Y gastric bypass has an increased FXR activity, which we know in animals can contribute to the benefits of bariatric surgery. So I will stop there and just summarize two major points that I would like to hope that you bring home. Is that the microbiota should be considered an important environmental factor contributing to host metabolism and physiology. But we really need to move from these association studies and try to find out, does the microbes really cause the disease or just mirror the disease? We need to find the mechanisms so we then can design intelligent human interventions and thus maybe use the microbiota to treat diseases. So I will stop there. Thank you for your attention, and just acknowledge the entire group who's really been contributing to this work. So it's a lot of people from many parts of the world who've been working hard on these different kinds of projects. So thank you very much, and we have some time for questions. It's crystal clear. There's a question up here.
My name is Jan Nyström. So thank you for a very interesting talk. Just a curious question. We have 1 kilo of microbiota. How much of sort of the calories we eat are used by the microbiota in percentage?
It's a really good and really hard question to address, because it also depends on what we eat. So if you eat a lot of sugar and fat, we probably absorb most of that in the small intestine, and not really where we have the bulk of the bacteria. Then all the fermentation we do, or they do for us, that's energy for them as well. And the short-chain fatty acids are related to other waste products. And the only thing we know in terms of that is that in my mouse experiments on a very fiber-rich diet, the germ-free mice have to eat about 30% more. Because they don't have the microbes to digest the fiber. And we also know that it's estimated that in a Western population such as us, about 5 to 10% of the energy we get from microbial fermentation. And if you then go to a more rural African societies where they have a more native way of living and eating more of grain-based diets, it's maybe up to 30%. So if you just eat like cassava, for example, we can't digest all the cassava polysaccharides, but the microbes can.
Interesting, thank you.
Okay. Then we save the questions for the general discussion later. So it's my great pleasure then to introduce Jens Nielsen, who's a professor at Chalmers University in Gothenburg. Received many awards, and not least the gold medal from EVA 2 weeks ago. And it's so, as I related to before, the microbes really have a lot of genes and a lot of functions, and they're really hard to assess. So what Jens is doing is trying to understand this, how we can assess the microbiota and make sense of all these signals.
Yeah, thank you. Thank you, Frederick. Yeah, so my entry into this this world was actually several years back. Frederick contacted me and said he was initiating a number of metagenome sequencing projects and whether we could assist in analyzing the data. And that laid the foundation for very fruitful collaboration, as Frederick has already alluded to. We already heard about the human microbiome, so let me just flip on. But maybe important to mention also that many of the bacteria and organisms, microorganisms that live in the human gut have also been isolated and their genomes have been sequenced individually. And this reference genome, as we often refer to them, are actually quite important for our understanding and also for interpreting and analyzing metagenomics data. And this is from a paper in 2012 where there was about 3,000 reference genomes available. This number has increased since then, and we now have maybe close to 10,000 reference genomes available, not all of them completed fully. So Frederick said that how do we study the human microbiome? About doing this by deep sequencing. So what is done is that the DNA is extracted from this microbial community, and then you do extensive genome sequencing. You generate very large data sets on this. I just gave some numbers here. Chalmers were very proud of their supercomputer, but the first time that we analyzed actually some of the data from these studies here, there was not enough memory in this, even though I had tried to convince them about that. That's because also most of the supercomputers in universities are set up for parallel processing, using what you typically do, parallel computation in physics and chemistry. Whereas when you do these kind of analysis here, you really need very, very large memories. But lucky-wise, we have proceeded with that and now have a very good collaboration with the supercomputer facilities at Chalmers. So what was done in order to develop and analyze these kind of data is to develop bioinformatics, and there you rely on a number of different processing of these kind of data. And one way to do is that you have these reads from the sequencing and you can align them to these reference genomes. And so you can basically say we have a lot of reads that are similar to the sequences in one of these reference genomes that I mentioned. And so therefore we can now say this species is actually available in that microbiota. Normally when we do that, it only allows us to align something between 60 and to 70, maybe in some cases up to 80%. So about 20% of the sequence reads are still not aligning to any reference genomes, and this is quite important actually. We can also do de novo assembly, simply as you do when you do a general genome sequencing, so you can begin to assemble them and build larger contigs, and that's also quite important. What is then, of course, important is that we want to get some biological meaning about this. So we want to integrate this with, for example, chemistry data on plasma and so on. We also would like to put it into context of biological networks and also correlate it with other kinds of information. So when Fredrik mentioned some numbers about genes, so we are basically beginning to touch and looking into a ecosystem of organisms that we don't really know of. So one could say we would like to have a reference genome of all the bacteria that lives in our gut. And so in order to do that, what we did was that we took sequence reads from several different studies, including close to 500 subjects, including several of our own. And then we were searching for genes, and not all of these genes have been annotated. We don't know what all of them are doing, and not all of them are in any of these reference genomes, as I said. But by de novo assembly, we can still identify genes. And then we come to about 12 million genes in our so-called gene catalog. The interesting thing about it is that this gene catalog is continuously increasing. The more sequence information we get, we don't really seem to saturate. But it should be mentioned, and I'll come back to that, that it's not really expanding necessarily much on the complexity of the system. Because a lot of the genes are unique to a single study. And if we look at it across, there's only about 500,000 genes that are really core in this case, 4 different studies. And so what is also interesting is that if we look at some of the core genes that are shared between the studies, these are also those that are most abundant. And when a gene is most abundant, that of course means corresponding organism, bacteria where this gene is present in, is also abundant. So that reduces somewhat the complexity, but still it's 500,000 genes. That is still a lot. One thing that we also looked into, Fredrik mentioned the different species or the different groups of bacteria here. We could see that there's a very large variability in these about 500 subjects here. You can see that when we just look at the Bacteroides fraction, that is varying very dramatically. We could also look into something about diversity. We can calculate a diversity index. It says something about how diverse is the microbiota in these different studies. And here we refer to them as China. That's the type 2 diabetic study from China that Fredrik was mentioning too. This is from an American study. This is MetaHit is from a European study, and the Swedish study is one of our studies that Fredrik also referred to. And interesting, you can see that the Europeans, and particularly the Swedes here, have a broader diversity. And particularly the diversity is low in the American study, and it's also lower in the Chinese study. And I'll come back at the end to talk more about the importance about diversity. So we've applied this bioinformatics pipeline for a number of studies. Fredrik mentioned some of them. Let me just give quickly some data from one study we did on type 2 diabetes, which is really where we started. It's about 150 subjects in 3 groups. Some of them were diabetic. Some of them were impaired glucose tolerance, in danger of developing diabetes, and some were normal glucose tolerance, what we would characterize as healthy subjects. And one of the reads that we can get out from this kind of analysis is that here you see again a similar plot as you saw before in Fredrik's presentation about the different bacteria here. And blue means that it is negatively correlating with some of these markers over here that were also analyzed in this study. And most of these markers here you would like to have low. Except for a few of them, adiponectin, for example, we would like to have no. HDL, you would also like to have low. But in general, you can see that these bacteria here are positive correlating with many of these plasma markers, indicating that they are kind of bad. And whereas you have some guys over here that are good, this is a good profile. And that's what Fredrik said, many of these are producing butyrate, and that's very clear from this study. But as I said, we are only harvesting, by aligning to these species, we're only harvesting about 60 to 70% of the information we actually have. So we developed also another concept that we call metagenomic clusters, and say, let's try to see if genes are co-occurring always in different subjects. So in other words, if these 3 genes are always reoccurring wherever we sequence in person 1, 2, 3, 4, and 5, these genes are likely going to be in the same genome. And so they are associated with one species. And so we can do that across and then find these metagenomics clusters. And some of these clusters are actually quite big. You can see as a few of them have up to thousands of genes. So they are likely going to represent actually full genomes. Some of them are small. Most of them are relatively small with maybe only a few hundred genes. But they are kind of the scaffold of a genome sequence for that species. And what is interesting is that there's actually additional information content in these metagenomic clusters. Because when we were running a model to see if we could find a profile of species that actually can predict whether you are in the risk or you are diabetic or not, we could not get a very good prediction if you just use the species information here, and the number here is area under the curve. I'm not going to go into the detail of that, but if that was 1, we would have a perfect prediction. 70% or 0.7 is not very good. But when we use these metagenomics clusters, we could increase this up to about 85%. And that's a fairly good prediction, actually. So it means that there is information content in it. The negative thing is that we need 50 of these. And that is pointing again to the extreme complexity, and that Frederick was also pointing to, that we don't really understand the mechanisms behind this. So that's really the focus of our work, is to develop models that can test hypotheses to see what are the functions and how are these different species interacting. And that can, of course, provide biomarkers for this, but it can also be used as design of interventions in terms of drugs, or which species are we going to develop for example, probiotics or for treatment of disease. So basically, we're taking, I would say, an engineering approach to this, where we are having mathematical models to kind of design how we can improve health of people in the future. And this is particularly necessary because of the extreme complexity of this system. That is very hard to generate one hypothesis. And we don't know that maybe we find species, bacteria, that are associated with healthy subjects, but maybe they will not colonize in that ecosystem because there's interaction with all the other hundreds of different bacteria that are existing there. So how do you model this system? Well, there are many different types of modeling used in engineering approaches. One is that you have a detailed description of the kinetics of all the growth of the individual bacteria. This is you could say, the most ultimate strength and the ultimate description, but that it has a major drawback is that we need a lot of parameters. And these parameters are very hard to get, and therefore often this simply becomes unfeasible to do. The other is that we use the metagenomic sequence information. I'll come back and touch briefly on that at the end. The other one is that we use information about annotated genomes. So we go back to these reference genomes and we build detailed metabolic models of that. And the last approach is agent-based modeling, which is a more heuristic and computer-driven modeling approach that is beginning to have some demonstration of power also. But the modeling approach that we are using is this concept of genome-scale metabolic models, and I'm not going to go into the detail of this here. But what we're doing is that we're taking, again, information from the reference genomes of these species, and we're collecting all information about all the metabolic capabilities they have. And so Frederick mentioned, for example, bile acid conversion, but it can also be that we know that this species here can actually degrade carbohydrates and it can make butyrate, whereas this species over here can consume butyrate, or maybe it consumes acetate and make butyrate, and another species is producing acetate, and therefore these two species can coexist. So that's the kind of models that we are building, and these models don't have any parameters. They're just having the stoichiometric information about how these different reactions are functioning in the organism. Turns out that when we do this, these models are still quite complex because they have thousands of reactions and thousands of enzymes, but they are highly connected, and that is actually quite important because that constrains the solution space. So some people who have been working on mathematical models would argue that it's not possible to do modeling without parameters. but actually it is to a very large extent because the system is really so constrained. And one of the reasons for this is also that it's a relatively anaerobic environment, and I say relative because there may be some oxygen in the gut, but it's very low amounts, and that has huge impacts on what kind of reactions can actually run in the individual cells. So what we are doing is that we are having these gut microbiota, we have metagenomics, we then identify individual species that are present, and we can then build these models for these individual species, and they communicate by secreting metabolites, maybe others are metabolizing these, and so we can have this community type modeling, but we're also looking into this pairwise analysis and co-occurrence of species. So we first did this in a simple study. This was a study that was performed. Data were generated in the lab of Jeffrey Gordon, where Frederick did his postdoc. So they colonized germ-free mice with individual bacteria, either a single one or a combination of two. And we then built these kind of models for these three species, for these three different bacteria, and we then could compare the model simulation with measurements in the fecal samples of the different organic acids that these bacteria were producing. And we found, assuming something about diet intake, that we could actually predict these numbers quite well. So this pointed to that we could have a causal relationship between the presence of individual bacteria and what metabolites are actually present in the fecal of the mice. More importantly, we could also look into what specific pathways, and one interesting thing is that when you have, what we found was that when we have two species, they actually not just interact by exchanging metabolites between them, but they also activate different metabolic routes in each other. And so this is quite important because clearly the community evolves and have a joint metabolism that is very different from the individuals, the two individuals. And so this is something that is very hard to deduce unless you have mathematical models to combine and extract information about it. So basically the workflow that we have is that we take data from human observations, then we do modeling, and then we can, of course, do a follow-up study in collaboration with Frederick on using gnobiotic mice. And so let me give an example from a study where we did modeling of data extracted from humans. So this was a study that was carried out in Paris, led by Karine Clément. And so what they did in this study was that they had about 50 subjects, overweight subjects, and they found that these overweight subjects grouped into two. The one group had a very diverse gut microbiota, and the other had a more reduced gut microbiota. And as you remember, the Americans had a more reduced microbiota, the Europeans or the Swedes had a more diverse. They referred to these as low gene counts and high gene counts, simply because when you are diverse, you have many more genes, and whereas when you are less diverse, you have less genes. And they did find in their study that low gene counts subjects had a higher HOMA index and other disease risk markers. None of these were diseased, but they were overweight, and they had markers that indicated that they were at risk of disease. So what we did was we then developed models for 5 of the most dominant species. So, okay, yeah, there are 100 at least operating microorganisms living in the gut. But we wanted to see, could we capture some of the effects by looking at just the 5 most dominant species. And we developed then a modeling framework to look into that, to see if we could model how these different species will interact and respond to changes. And what they did, I should mention first, we then validated in Frederick's lab the performance of these individual bacteria. So they were grown individually, so the model was if compared with experimental data where we looked into the uptake of different amino acids, production of different short-chain fatty acids, and so on. So the models were kind of validated individually first. The models were also evaluated by looking into consortia, by 4 species growing together, as still in the test tube, so still not in any human. And again, we had good consistency between the experimental data and the model prediction. So that was kind of a validation of the modeling concept. You could say that this seems to work. But when we go and are going to describe what is happening in the human system or the human samples or individuals, it's of course much more complicated, because we are having to translate information about what diet they are having, the individuals. So we had to develop also a diet algorithm where we took information about what are the amino acid composition and other micronutrients in a number of different foods. And we can then use that to calculate when you have, for example, information about how much bread or potatoes and so on the individuals were recording to have. And they were, in all these subjects here, they were put under a 6 weeks weight reduction diet. And so they were monitoring, and you can see there were some changes in the diet also between the low and high gene counts. And these are the colors here indicate the changes. So, for example, they had an increase in cheese, they had reduction in bread and carbohydrates in general. They had increase in fruit and vegetables here. But most of the diet's components were reduced. And the color code, there are some variations here because these are from records from each individual. So there are some variations, but we had this for each individual person information. We could then take this together with information about what is the prevalence of these 5 different species here over time, and this was at baseline, and then after 6 weeks diet reduction, weight reduction, and this was from the 2 groups, high gene counts and low gene counts. So we have in total 4 groups here. And one thing that we could then find was that the model could predict that what happens when we go from the baseline and we measure the amino acids, a number of different amino acids in the fecal sample, what happens when you go from baseline to after 6 weeks on being on a diet with reduced calories, you get a reduction in this. So all of them are going down on this axis here. Interesting, what is happening is also, if you look on this axis here, and here really the baseline here is 0, what is happening is that over here at the baseline, the low gene counts have higher levels of all these amino acids in the fecal samples than the high gene counts. But this changes and almost equilibrate, and actually maybe even flips over after 6 weeks diet reduction. Now, what is this? Is this consistent with measurements? Yes, when we compared that with the correlations, we could actually find that was quite consistent with the model predictions and the measurements also. This is also seen actually if we measure the amino acids in the blood or in plasma samples. We see the same thing, that again they are shifting from this side of the figure here down, but also to the right here. So in other words, the low gene count subjects, when they go on a reduced diet, they actually get lower levels of their amino acids in their plasma as well as in their fecal samples compared with before, so they are responding stronger. And we believe that this is because of this reduced gene count or reduced microbiota, that they have a different capability of actually metabolizing their diet. This can have a number of implications because the amino acids can have a number of different functions, of course, both in terms of energy harvest, but also a number of regulatory functions. So then let me end the last 5 minutes to talk a little bit about the interaction between species. So here again, we took these 4 different studies. I think maybe it comes to close to 700 subjects, and to identify key species abundance. And so what we did here was that we ran a number of computational algorithms, but let me just simplify to say we are looking for species that seems to co-occur across many subjects. If we are going to be very strict on this and say these two species should co-occur all the time in all 700 subjects, then we would find nothing, because there are variability. And sometimes also, if you are looking into species comparison, whether species A and B are present in all subjects, maybe B is exchanged with C in many of the subjects, but B and C have very similar functions. They can actually do very similar things, because there are pretty large diversity in the bacteria, but still also many of them do similar things. So that's the kind of analysis that we are doing. And we were particularly interested to see if we could identify so-called core species and generalist and specialist. And so this is the kind of analysis that we find. So this is the core occurrence network for all the subjects. But when we compare that, and so an arrow indicates that these two species are co-occurring, and you can see that there is a lot here, a lot of species that seems to be co-occurring in many subjects. And so they are probably important to be together. This is quite different if we look, for example, in just the Swedish study, and we took that out just as an example, that then you start to get different patterns. And this is just to illustrate that that may change in cohorts as well as also in health and disease. So, but in the grouping of the many different species, we find we group them into core. These are the most abundant species, and we have a cutoff that they should be about more than 1%. Generalists, they are less abundant but present in many subjects, and then we have what we call specialists. And what you can see here is that we find only about 113 core species that are present, but they are also really the most abundant when you see here. You can see here some of them are, I think actually it was 1 per mil that is the cutoff, but you can see also some of them are up to representing about 1% of the abundance of these species. So you have a set of core that are really relatively abundant. So coming back, we often talk about this with thousands of different species, but really it is a much reduced number if we look at it from this. We have some generalists and we have a few specialists here, but if you look at all, you can see the number of species is approaching 1,000, but most of these are really, really low abundant. And we don't know if they're playing any functions. It could be still that they're playing a function in terms of immunostimulation and so on, but in terms of metabolism, they're probably not playing a very important function. So these about 100 species, they have a wide phylogenetic diversity. Let me not talk about that. But just illustrate here these clusters of co-occurrence. And here you can kind of see that you have a number of these core species here that are interacting. But here you can begin to see that these two species here, for example, they cluster together. That means that they are often co-occurring. But they are also connecting all these species here with all these species here. And so we refer to these as keystone species because they're kind of important. You can imagine if they fall out, this complex here will fall apart, and maybe these species will not be able to coexist in this, and suddenly you will get loss of this whole set of species here in that microbiota. And then, of course, if this plays an important function, that can have detrimental effects. Not going to go into the details of this, but just illustrate here that we believe this has relevance for disease, because when we look, just zoom in on the about 150 subjects in the DIVA study and look at the normal glucose tolerance in the type 2 diabetes subjects, we do see quite distinct differences between these graphs. We don't know what the functions are yet, but we are now building these detailed models for all these species so we can look into what are actually the functional activity, metabolic functional activity in each of these subjects, and that can drive us forward to begin to build models for larger communities and also actually what drives the connectivity between these species here. Let me just mention right at the end here, we've also been looking into this on association with malnutrition. So a number of different studies have been looking into this in different children, and where there seems to be really also differences in relationship between the firmicutes and bacteroides here. And we have been building models, and what it seems to be a strong indication of that the malnutrition of the children, particularly in Malawi and Bangladesh, where many of these studies have been performed, are very much associated with the capability of these bacteria to actually capture some of the essential amino acids. So they it may go together, that they are important fermenters, but really in a protein-reduced diet, they are also important in terms of generating and producing these amino acids for growth of the children. So hopefully I demonstrated to you that we can use metabolic models to get information about the dietary changes in the uptake of small metabolites. We can have this core current analysis to identify clusters of species that coexist. And hopefully, going forward, our analysis will allow to design probiotics that can, through coexistence, will stabilize the presence of a healthy gut microbiota and hereby revert dysbiosis. And again, let me acknowledge the people who did much of this work, and you for your attention. Thank you.
Thank you, Jens. Is there any questions for Jens? So,
I am Radetzky. I am a simple economist, and the presentations I have heard are far above my head. However, I may have misunderstood, but correct me about it if so. I understood you to say that you were using mice that were germ-free for some of the experiments that you were performing. I was under the impression that germs have to be there in the gut in order for the animal or human to survive. So how did your germ-free mice survive?
Well, it's actually his. But Fredrik, you could maybe better answer this question, I think.
It's germ-free mice survives. They need some extra vitamins and minerals, but otherwise they survive just fine. The problem they have is if there's a bacteria introduced to them, like a pathogenic bacteria causing disease, they are much more sensitive because they don't have the other microbes to protect them. They also need to eat more, but they live longer. Would humans survive? They probably would survive, but it's not going to happen because you get bacteria through the environment. So you can't do that experiment. It's impossible. But you can get animals to survive under germ-free conditions. Actually, one of the main problems is that animals get problems of detoxifying environmental pollutants. So what you see is that the germ-free mice dies much easier if you puncture the cecum, for example, the gut, and then there's more toxic compounds coming out. So the bacteria are good for you.
Can I ask a question about key species? Because I'm a doctor, I will show some clinical data, and I'm always thinking, how can we use the insight that brilliant engineers like you give us? So can you speculate? Can we also use the keystone species to knock them out in order to cure a disease? Because you're saying, exactly. It's like playing like bowling, right?
Exactly.
So can you say something about it? What your envision is about a therapy or?
Yeah, so this is a very good question. So this you could definitely also do if you can find targets that are specific for those species. But you may be able to do that. You may be able to find parts of metabolism that is essential for those species, and then design drugs to target, for example, those particular enzymes. It can also be that you can have very directed antibiotics. Let's say maybe some of these keystone species fall in a certain class of organisms or bacteria that you can kill with targeted antibiotics. That's probably going to be more difficult. Maybe what is more likely is, for example, antisense silencing and stuff like that. But that is hard for bacteria still. But that would be more directed.
Okay, so we heard the biologist view, we heard the engineer's view, and now the medical doctor's view of the microbiome will continue. So welcome Max Nudorp from Amsterdam Medical Center in Holland.
Okay, it's working. So I'm from beautiful Amsterdam. I have to say I'm always impressed by what all scientists can present us about engineering and modeling. But I am still practicing doctor in the clinic, and I will take you in the next 25 minutes about what is the relevance for us as humans, these microbes, and can we really dig into it and get some new medication out of it, because then I guess in the end we're really doing, for humans at least, something good. So you might know from lay press that what Frederick said, if you open up a newspaper, you read about microbes everywhere. There are much more microbes on the planet than humans. And I think so that's good to realize. Some of my slides, by the way, are redundant. I'm going to skip a little bit over it. But this one is certainly not redundant. Many of the research that is published in these journals are in mice. And I have to be straightforward with you. Of every 100 genes that have been found in animals to cure diabetes, only one is relevant in humans. So many studies that are done in mice give us a good idea about how a system works, but they're not so relevant to understand the human disease, and that is because the human, of course, has much more of counterbalancing reactions. We have a different immune system, we have a different diet. So animals are more controlled, for example. A human usually lies about what he's eating. When they come to me in the clinic, they tell me, "Doctor, I eat almost nothing and still I gain weight." That's the thing. So we call that confounders. So animals have no confounders, humans have large confounders, and especially diet and medication are important. So I think that's one good thing to realize. And the other thing, what the other previous two speakers said, is that when we're all here in the room, we have maybe similar but different microbes, but it's really different how I eat my hamburger and I digest it into metabolites. Then when Frederick eats his hamburger, because he's from Sweden and I'm from the Netherlands, we may be eating more cheese, I don't know, or more dairy products. But these metabolites, they go into the blood. And it's not the microbes that go into the blood. We don't think that that's the case, but it's the little parts of the food that drive parts of our brain and the heart and the muscle. So that is important. It's also very important that microbes have been around always. So you're not, this is about debate, but I'm a believer that we're not born sterile, so that in the womb, when you are growing there as a fetus, you're exposed to bacteria, because you need to get your immune system, when you're protected there in your mom's belly, has to know, okay, we're coming to an environment where not everybody is friendly and there are microbes that want to attack us. So you try to, at least in the womb, you're trying to be trained for that. And I think the other thing which is important is that not everybody develops the microbiota in the same trajectory. If you're taking antibiotics as a child between 0 and 2 years, it might be much more harmful and driving you into obesity at later stage than when you take the antibiotics at 4 years. And I'll come back to that later. So we have a little bit of a fingerprint. Medication is important. Not so much antibiotics is good for you, but also simple things like how you are born are driving it. And I think we know from large studies here in Scandinavia that if you are born by a cesarean section, that you are more prone to become obese. And the feeling we have is that when you're born by a cesarean section, you're put on the skin of the mother, and you're not going through the vagina of the mother, where the microbes there, which are covering you, mimic the gut. And small studies have shown that indeed, if you are born by a cesarean section, so by directly taken out by the surgeon from the womb, you have a different microbiota in your stool. Vice versa, if you take breastfeeding, and in the Netherlands we take a lot of breastfeeding, I think in Sweden it's the same, you protect your child from getting obese. And this is driven by the fact that, well, the milk from the mother has bacteria in it. They dwell in the nipple area, so the child suckles it and ingests them. And on the other hand, bottle-fed babies, the bottle feeding is usually quite sterile because it has to go via specific rules. So all these things, little things, might be important, and it's just at the start of your life. But people let's say, it might be very intriguing to think that at the start of your life, you're basically either determined to be obese or not. I think that's not very much the case, but it's interesting to think about that every little thing, like little swapping of the butterfly in the tropics might drive an volcano somewhere else. There's these kind of things that you think about youth effects in adulthood and driving diabetes. And obesity and diabetes is a big thing. It's expected that 1 out of 5 adults will have obesity and diabetes, so it's a lot. Much is driven by what we're eating. So I'm not going into microbes or how they are driven by food, but just showing you simple things like fructose. We did not eat that much sugar or fructose when we were in the Middle Ages, but sugar and fructose are addictive. So they are cheap to produce. If you take them, you feel full, but you feel full only a short while, and then you want it again. It gives you a kick. And what you can see is since the 1900s, we in Europe have been eating more and more sugar, but in the US, since the 1700s, they're eating much more sugar. And it's just because their natural harvest just has more sugar. But we're talking about 33 kilos of sugar per year. And sugar, as I said, is different than fats or fibers in driving food intake. So only what I wanted to show you here is that if you people usually lie about our diet, as I said. So when you really want to study it, you lock them up in a room like we do here. We wait for 7 days, and in 7 days we provide you with a healthy diet, or we provide you with a cafeteria diet. And then, indeed, you can see in students, this was done in the US, that if you give people a fat diet, you make the bad bugs bloom and you get less of this butyrate. If you give people the good diet with the fibers and stuff, you get the good bugs to bloom and you get more of this butyrate. Another thing which I wanted to discuss with you is antibiotics. It's under debate, but I just want you to think about it, because when I prescribe antibiotics, and I do this a lot because I see a lot of people with diabetes that have food problems or infections, and I treat them because I don't want them to be killed by the bug. And since we have had antibiotics in the world, we know that less people are dying of infections. Fortunately, I would say. So Alexander Fleming really got the Nobel Prize for the right thing. But in parallel, we see an increase in diseases that are, at least we think, associated with these are autoimmune diseases which are linked to bacteria. For example, we get more Crohn's disease, which is a disease where your bowel inflames, and we get more type 1 diabetes, which is a disease where your pancreas is knocked out. So the thing is, does it have anything to do with our antibiotic use? So there's one person who knows a lot about this, and he's called Martin Blaser. He lives in the US, and he did a very interesting human experiment. He and his wife, they went to the Amazon Indians, Amazon, where people live that never been exposed to our Western lifestyle. They took some fecal samples of these Indians, and they compared it to Caucasians in the US with their bad food. And they estimated that these people in the US lost about 30% of their whole bacteria in their gut compared to the Indians that are still living like in the Stone Age. And he believes, and I think it's true, that if you believe that we get our microbes from our parents and from daycare, and we're breastfed and all this stuff, and since the Second World War, we have been exposing people to antibiotics and to more increased hygiene, so less bad bacteria come in, your body is less trained. And I give 50% of my bacteria to my child. Each generation, you lose your diversity. It's a simple calculation. I don't know whether it's true, but it's interesting to see these pilot data popping up. The other thing which is quite interesting, especially for me as a doctor, is that antibiotic use is mostly prescribed in the poorer parts of the US. I know how it is as a doctor, because you have 5 minutes for a patient, they have a cough. You rather prescribe a prescription, because then you will not be sued, because you do something. But the thing is, if you prescribe antibiotics without a good cause, you might also kill the good bugs. And Martin Blazer says, interesting, in the parts where most of the antibiotics is used, most of the obese people are living. Now this is called confounding, because obese people or poorer people go to the doctor more often, they demand more medication. But if we think that this might be true, if we think that antibiotics is true, you go to registries, and what you just do then, and can be done again in Scandinavia, say, okay, we just count the amount of antibiotic courses that are given, we just count the amount of babies that are getting obese, and then what you can do is that and knowing that in the US a baby gets, of a child until the age of 7 get 12 courses of antibiotics, whereas here in Europe it's only 2. Here you can calculate that the numbers needed to treat, which is meaning how many people do I have to treat with antibiotics to get 1 obese child? It's 40. And these are registry data, right? So it's not lying. It's just about if you treat 40 children with antibiotics because of their cough and because of their worried parents and you don't want to be sued, you might end up with 1 child that is obese when you prescribe antibiotics to 40 of these children. We need to think about that. So if you like this, you could go to the Missing Microbes book of Martin Blazer, where he describes all of this, stop the killing of beneficial bacteria. It's more of giving you some food for thought. The major thing what we have is just environment. So it has been known for 45 years that lean dogs have lean owners and obese dogs have obese owners, just because dogs eat the same, but also the owner of the dog says, I'm not going to walk you because I'm sitting here in my chair and I don't have time to do my exercise because I'm watching TV. Now the interesting part is that we share in our households our bacteria. So when I shake hands with you, I expose some bacteria of me to you. The question is how much you take of those bacteria from me. But we know that dogs have a similar bacteria in their gut than as the owners. And it might be 50%, but not only in the feces, as you can see, but also in the tongue and in the hand. So Frederick showed you that microbes are associated with many diseases. I know that diabetes is associated with many diseases, but this is for me, as a doctor, why I went into the field together with Frederick and together with Jens. Because my idea is this: if you know that you have a disease like diabetes, which takes on for 5 years before a patient comes to the doctor and says, I'm ill, then what you want, at that time, this patient is way too far with his disease, so the body is not flexible anymore. The disease is so prone, you have to treat them on medication or you have to treat them on insulin. And what I want, what I hope with the microbiota research is that you can pick out these people much more early before they are failing with their pancreas, pick them up when they're still just happily living around, and then say, hey, I'm telling you as a doctor, you have a risk of 3-4 times higher of getting a disease. Maybe now we get you motivated to do something about diet. So I think that's the important part. This one I will skip. This is what Frederick did. And we will go to one thing which is also important. This is a hype. This microbiota is a hype. So I call that the pink elephant in the room. Because there's, as I said, many of these animal data that are generated are just plotted on human disease. So they found an animal model of autism and they say microbes are altered. So the same must go for humans. I don't think that is the case. The other thing which is quite important is that we need sample studies of like the size of Stockholm or the size of Gothenburg to have like 1,700 subjects per patient group to give a good idea, and not the hundreds that are published now. But the question is, if we know that microbes are different, can we restore it? And that is something that I will tell you now in the next 10 minutes. We do fecal transplant, which is basically very, very easy. I have to admit, we found it out, and I'll show you that in the next slide, just by Googling it, so it's not rocket science. And fecal transplant, what it does here, you can find it in Chinese handbooks. in the 4th centuries before Christ, where the Chinese doctor said, if you have abdominal cramps, take feces from a baby, a healthy baby, which was called yellow soup. It has been used in animal care it's as filthy as it is, but you can imagine that with all the stories we heard from the previous speaker that good bugs might be in there and you are missing the good bugs, the best you could do in those stone ages is to use it and ingest it and hope that it will come there. So many, as you can see from this slide, many of the research has been done way back before I was born, but here in the 1980s, then the field is coming to bloom because we can sequence these bacteria and we can culture them. So this fecal transplant, of which is just giving stool to another person, has a pro because you look at everybody as his own control, which helps you to keep out the noise. But the contra is that we're infusing much more than bacteria. So there is some ethical part to it. We've shown for chronic diarrhea that it really helps, and we made the New England, which is a quite good journal for it, that if you treat donor feces, if you use donor feces to treat chronic diarrhea by overgrowth of one bacteria, you treat 90%, you cure them, and antibiotics, it's only 30%. So that sparked a big boom of research, and if you go to these websites, you can find 190 studies studying fecal transplant in any disease. It's very easy, as I said. You just ask a patient, of a healthy subject, to come to the hospital, put the feces of those donor in a blender, and then give it either via the stomach or rectally. But if it's so easy, people come up with their own solutions if the doctor says, I cannot help you because this is too dangerous. So I've had patients that came to me and said, can you treat me? And I told them, well, I don't know anything about autism. I cannot help you. Then they came up to me 3 months later and they said, I found a tutorial on YouTube, 30 minutes, where they asked me, take a blender, take some water, take some stool from some donor, and just do it yourself. And that, of course, can be very dangerous. The other thing we have is that in the UK, you have a big commercial transplant clinic where people go for £5,000. What is that? About 50,000 Swedish krona. And you can be treated for anything you want, as long as you sign the contract that you don't sue the doctor that is doing it. But we try to be scientists. That's why I'm really just a simple doctor. I need to collaborate with smart people that help me once we do these interventions to see which are the bugs that we need to treat. So at the AMC, we're doing many of these fecal transplant work for many diseases, fatty liver disease, but also anorexia nervosa. I have family members that have anorexia nervosa who cannot gain weight because their brain is so strong in suppressing their food intake. And I was wondering, can you just give obese people's feces to these anorexic patients, the anorexic girls, and maybe make them eat a little bit more because the gut talks to the brain and tells them to eat more. This I can skip, but what I want you to know is that we have to do very sophisticated measurements with all kinds of label isotopes and all kind of insulin to see what the effect of microbes is on glucose metabolism. And if it has to be so sophisticated, we usually don't pick up the effect with an oral glucose tolerance test. It means that the effect is not so large because you have to take very sophisticated measurements. So I don't think microbes are going to cure diabetes. But they do something very, very interesting. And these are the final 5 slides of my talk. It's once again all these plots with the colors, but we're not going to look at it. The only thing I want you to realize is look at this figure where we used the feces of healthy people and they were very, very good in their glucose metabolism. And we compared it to patients at baseline who had diabetes. And you see that the curves are lower. These people have an impaired or disturbed glucose metabolism. If you give people their own feces back, the curve is the same, nothing happens. But if you give them the feces of this donor to this patient, you see it goes up, so you improve it, but only temporarily. And not everybody responds. And then people ask me, how big is the effect? Did you cure diabetes? And then I told them, well, if we give people medication for diabetes, if we give people metformin or if we give them new medication, you can only do about 20% improvement. Same with the feces. So we're not doing a beneficial thing in largely just wiping out diabetes, but we're just treating diabetes in a different way. We were reproducing it. We showed that it didn't take long, only 6 weeks, then the effect is gone. But we found something else. We found, and this is I think we're almost there. We found in this study that if you take a donor, we can predict based on the machine learning of Jens who is going to respond, who is going to accept these bacteria of the healthy person, and which patient is saying, I don't want the bacteria of the donor, I just keep my own bacteria. We can do that with all kind of interesting techniques, but this, I think, this one figure shows better than any other thing. We published it a year ago. Look at all the colors. The colors are bacteria that are changing, and it's one donor that is used 3 times for this, and this patient. And if you see an orange color, it means that the patient really loves the bacteria of the healthy person, the patient. So they engraft, they find a new home. But if you see a blue color, it means that the bacteria that are coming from the donor are just kicked out immediately, and the patient keeps his own bacteria. So you can see that somebody accepts very much, the other doesn't. I think it's interesting to notice. So then, where does it leave us? I think it will leave us with this as doctors. We're not going to do fecal transplant clinics. We're not going to set this up. What we want is one of the first studies from the surgeon that did the first fecal transplant in the US. He said it's much better to give pure cultures, pure bacteria, keystone species, as Jens said, that are missing. Give them back to patients and see if you can improve health. We're trying to do that. It's called reverse engineering with Frederick Beckett. But the thing is this, getting an idea about how big bacteria are playing a role in the disease, we will find out that with the overgrowth of bad bacteria, there you can cure anything. But if you go to complex diseases, which is obesity, the microbes are not driving this disease. They're just like a rocket that goes to the moon. You have two motors besides it. That's what the microbes are doing. They're keeping it stable, but they are not really, if you kick out the bad bugs, you're going to prevent diabetes. I don't think it's that important, unfortunately, but we need to focus on it. So my conclusions are that microbes are important, and it's worthwhile to go for as a doctor, because diabetes is going to be a major disease, and we only have medications that can only partly treat the disease. But we have not proven that microbes are really causal, are really driving this disease, not for any disease, except for the chronic diarrhea by C. diff. And this FMT, this fecal transplant, helps us on an ethical verge, because, as I said, we're infusing much more than just bacteria. It helps us to treat diseases, and it might help us to find the new keystone species, new probiotics that we would like to identify and then give as a doctor to a patient to cure him or temporarily cure him. So with that, I would also like to thank my group, but especially Frederick, who really brought me into this field, I think 5 or 6 years ago, or at least got me in a more scientific way. And I'm open for questions. I saw I did it exactly in 25 minutes. Thank you very much.
Thought we'd do like this, that we take some questions for Max, and then we will have all 3 of us up here if there's anything you would like to ask us. So questions for Max, please.
Max, could I ask you a question? Staffan Jonsson is my name. About these products now on the market where they say they have good bacteria in them. I mean, a lot of milk products and so on. Are they helping people? Or?
Yeah, it's a very good question. So the question is, are products, and I think you refer to either probiotics or just products with high fibers or dairy products. So in the Netherlands, we don't have a history of prescribing probiotics, so I was very skeptical when I came into the field. But it's called meta-analyses, and I was very surprised to see, for example, when I prescribed my patients antibiotics for their wound, and you it's basically shown that if you give these people probiotics, that they have less diarrhea, shorter term diarrhea. So that for that field, it's covered. It's much less clear for diabetes, or like Frederick said, with bread that gives you better glucose. There we go into not one size fits all. I skipped it for time, but there it will really be that some groups of patients will benefit and some will not. So in my opinion, there are some diseases like trial diarrhea or necrotizating colitis, that one, and diarrhea induced by antibiotics or traveler's diarrhea. There these products may help and are proven to help. But for other diseases like IBS, so it's irritable bowel syndrome, or diabetes, I think the picture is less clear, and it will be that some groups are responding and some groups are not, and that's something we have to do. We have to look at this in a total different angle. Larger studies, longer follow-up. Yeah.
So Max, what do you think would be the best treatment option then if we base it on microbes?
Yeah. I have an idea about that. So in my dream, you have a cord blood bank, right? So when a baby is born, commercial companies say we store the cord blood, which has pluripotent stem cells in there. So when a baby gets sick 30 years later, we go to the freezer, take those cells, and for example, cure the baby from his leukemia with these cells. That is not science fiction, it's really happening. So I think what we should do is do these stool banks. So when you become 18, you have this, I don't know whether you have these biobanks in Sweden, but in Copenhagen, they have, in Denmark, they have these biobanks. So when you become 18, you're asked to ship a part of your stool to a biobank where it's stored and frozen. And when you get sick, for example, or you get antibiotics, you can use that part of that stool to cure yourself. It's much more ethical than what we are doing, going to healthy people and who are unrelated and infuse those feces to these patients. What I'm suggesting is we're not at, I don't know whether in the next 20 years we will be able to grasp which bacteria are really important and who's going to benefit from it or not. So the first answer to that question is let's just store now and save it for a rainy day, the stool. The other thing which is very appealing to me is that if the key species idea is true, I think it's true, but it has to be shown in the healthy population and not so much the disease population. It might end up that you come to me and you tell me, doctor, sequence my stool, I still feel healthy, but I would like to know which bacteria I'm missing. And then I come back and I say, I see that you missed 10 of important bacteria, and you go to a company that just has these bacteria, these 10 bacteria on the shelf, puts them together and provides them to you into like a half a year or something as a pill daily. But this is very expensive. This is not what a pharmaceutical company usually likes to know, because it's tailor-made. It's not one pill like a statin that you give to everybody. So I think those are the two flavors we have, looking at it from a doctor's angle. But it might be that I'm wrong and that we go back, for example, to phages, which were important before antibiotics came up. It might be many things, but this I think is...
So one thing that bothered me was that you said that the field we're working in, diabetes, has no chance to be successful.
Mm-hmm.
So what disease...
I told you it was less, we have to work hard to be successful. Yeah.
Okay, so if I don't want to work so hard, what disease should I work on?
C. diff.
I bet everybody's doing that.
Yes.
You have to give me another thing.
I think inflammatory bowel disease is the next best shot. So if we go to this, we really digged into the literature to get this pyramid, right? So of complexity. So causal C. diff, you have multi-resistant bacteria, for example. I give a lot of antibiotics, and sometimes a patient comes back and I swab him and I see that the bacteria are resistant to these antibiotics. It's called an ESBL. And with a fecal transplant, you can wipe out the ESBL. So you can start all over again with a patient, basically neutralizing it and say, oh, we can use this antibiotic, which you were resistant before, but now we can use it again for 5 years and we cure you. Those are the ones that are most prone, I think, the next 10 years to be helpful, and IBD, so inflammatory bowel disease. But the diseases which are more complex, like where the brain is involved, diabetes is a disease of the brain and the gut and the liver, it's the more complex you go, the more diverse the disease is, the more complex it will be. So I would bet you on multi-resistant bacteria that can be killed by these interventions, so we can keep on giving cheap antibiotics and not these expensive ones, and inflammatory bowel disease, I would say. This is a simple doctor's view.
So are there any more questions for the simple doctor?
There's one in the back.
So all of you are sort of saying that diversity is good for us. So what is actually happening when you're giving antibiotics broad?
Yeah, it is very interesting. We studied it. For the sake of time, I didn't show it. If you give antibiotics that are aimed at gram-positive bacteria, those are the ones that usually live on the skin, but they also live in the gut. If you do that, you screw up the whole system. It implodes. So if you come in with antibiotics aimed at gram-positive bacteria, you get lower diversity, and the whole system implodes. So it means to me that the gram-positive bacteria are one of those keystone species. If I come in with antibiotics against gram-negative bacteria or anaerobic bacteria, not so much happens on the diversity, as far as I know from literature, which means that probably these bacteria don't kill all of these gram-negative bugs, and the gram-negative bugs that survive, they take up this part. So, but antibiotics do have an effect on diversity, and I think the most compelling evidence we have is that if I give 1 antibiotics to a person, has been done by David Relman in the US, 3 days of antibiotics, and even 3 months later, the microbes are not fully recovered. So the effect of short-term antibiotics is very long-lasting, and you can still pick it up, and then realize what I said, that the children in the US have 18 courses of antibiotics before they are 7 years old, and in Sweden and in the Netherlands about 2. If the effect is so long-lasting, each time you might lower a little bit of diversity, but if you give 12 or 18 courses, you will do something. So there is an effect that's small, but it's long-lasting.
I think that's a very important note, because what we see more and more is sort of like a boxing game, that you may survive 1 or 2 hits, but if you have too many hits, you will eventually lose the diversity.
Yeah.
I think there's only one disease where there's been associated with an increased diversity, and that's type 1 diabetes.
Yeah.
So what's your view on type 1 diabetes, which is opposite? Because
I don't know whether everybody knows what type 1 diabetes is, but it's really I'm an endocrinologist. If I have to see one of the most devastating diseases we have in that part, except for cancer, in the pituitary, is type 1 diabetes, because you get it usually as a child. You're admitted to the hospital, and then you have to inject or put insulin in yourself your whole life. And we don't understand the disease. They have come around with bazookas of anti-inflammatory disease treatments. They have come around with even chemotherapy and stem cell transplants to wipe out the immune system, and nothing helps. The only thing that helps is giving back new islets of Langerhans, which are from the pancreas, and they are also killed by the immune system. My idea is that microbes, this is my personal feeling, so the microbes those patients have are not driving the disease, they are protecting the patient from getting even a worse disease at all. And maybe that's why we see this more increased diversity, because these microbes do like a buffer. So they help to keep the immune system in check and not driving that disease. But that is my feeling, and we have to study it. But I agree with you, this is the only the only part where we know we have higher diversity in the disease. And on the other hand, it's such a devastating disease that we only have a medication cure for, but we don't understand how the disease is driven.
You mentioned treatment. You mentioned treatment with fecal transplant in anorexia patients, but you didn't comment on the outcome.
No, because it's ongoing. But and the same goes for. So it's very nice of you to ask, because it really was in the middle of the night. I woke up. We had a night shift, and I was thinking, so why am I working on obesity when everybody tells me that these microbes which we have in obese patients are only driving maximum amount of food uptake in the gut? So why not taking it 180 degrees different and use the feces of these obese healthy subjects in people that need more of those uptake in the gut? So that's why we went 2 years ago to patients with cancer cachexia. So patients who have cancer and get chemotherapy, they eat less. It's because of the inflammation, because of the chemotherapy. Frederick already alluded to that part of the microbes helped for how the cancer medication works, so that's part of it. So we started a trial, it's almost finished, where we gave the feces of obese, healthy people to cancer patients just to see if they would keep a stable weight and not lose weight. And the same goes for anorexia. But if you do this, you have to take two things into account. You have to do a placebo arm there as well. So half of the patients that you treat get their own feces. So is that ethical? You have to do it because otherwise you're having a placebo effect. And the second one is you cannot do these trials and then say, hey, can you just fill out how you feel just on a questionnaire list? You have to do more. So you have to do brain scans in the anorexia patients, or in the cancer cachexia patients. We do CT scans to measure how good are they gaining weight based on more fat, or is it muscle mass, or all this stuff. So it's a lot of time, a lot of money, but I think the only way. And my feeling is, based on a few case reports, that this might be even more efficacious than trying to cure diabetes or obesity by the microbiome. But once again, maybe in 2 years we know more.
You have discussed the use of antibiotics for treatment as a doctor.
Yeah.
But today a lot more antibiotics are used in the farm industry.
I know.
For breeding animals, etc., and speeding up the growth there.
Yeah.
How does this affect us, and how does this affect
Frederick,
the
do
overall
you want to take that
microbiome?
one? It's so Martin Blazer has asked this same question, and what he did is he used these very tiny amounts of antibiotics that are used to grow cattle more quickly. So, but it's weight gain in cattle. It's not only fat; it's also muscle, and that's not what we think we see in humans. In humans, it's more fat and not so much the muscle that grows. But anyway, so he investigated it in animals, in mice. He gave them these tiny amounts of antibiotics that are used in the food of cattle. Just in the Netherlands, we are the worst in that case. We have no resistance in humans for antibiotics, but we have major resistance against antibiotics in our pigs and other stuff. And there he could reproduce that if he gives tiny amounts of antibiotics, he drives animals to grow faster, get a different bone mass, get a different muscle mass. And if you then think that part of those antibiotics, maybe then 1/100 of that, ends up in the meat and it comes into you, it might be important. The thing is how we should prove that. You have to go to registries again and ask people questionnaires about eating. But then what I showed you is that people usually don't really recall very well what they eat. It's not so much about lying, it's about recollection. So I think there might be an interaction, but it's not fully studied. The only thing we know is giving little bit of antibiotics alters microbes, gives a different drive of growth in animals, and whether it's in humans.
I think also one part in animals is that you protect against pathogenic bacteria because they are living in pretty crammed quarters, and you reduce.
So they just die less quickly, or something.
Less infections. If you have less infections, you gain more weight. We have a question in the back.
Yes, I'm wondering, there are quite many recent papers now on immune checkpoint inhibitors and cancer treatment, and that's quite exciting. Where do you want to put that on your pyramid, if that's a possible future?
So the question is this. So there's been a couple of papers, not many, so it might be lucky shots. I don't know much has been reproduced, but what has come out is that if you give specific checkpoint inhibitors, which, for example, people with melanoma used to die really quite quickly, you come in with this new medication and suddenly they survive 3, 4, 5 years. The old president of the US, Jimmy Carter, everybody knows he's on this medication. He's still alive with a metastasized melanoma. The thing is, these medications have side effects, and about 20% of patients that use it gets IBD. They get inflammatory bowel disease. So there is an interaction with these medications, the immune system, and the microbes. But it's also been shown that, just like metformin, if you give this medication, the bugs are the first to see the medication. It's not the liver. The bugs are the first in the gut that see a medication and might alter it. So I have a feeling that many of the medication work that we see might be driven by how the microbes are processing it in the gut. And I think the best example, which I feel that might be important, is, for example, some people on statins, cholesterol lower, they have muscle aches and stuff. It might be that this is a relation of how we take up this statin, and how before the statin is taken up by the gut itself, the microbes are changing the molecular, for example, a change of that medication. So I think it's very interesting. I know many studies are there, and we are doing it vice versa. So we're doing it with these checkpoint inhibitors. We're doing studies in combination with FMTs. Yeah. But I would not be surprised. I keep on stressing it that many of the studies that I present you are negative because it's a black box. You put in something like a feces from somebody else, and you hope to see an effect. But it will get more sophisticated when we go along the way. When the same is for these kind of causality or large studies, it will get more sophisticated because we get these large cohorts of 2,000, 3,000 patients per disease, and then we can correct for all of these confounders, and then we will get there. But it's a fascinating field because it means that they are involved, these bugs, in almost all of our metabolism and health and disease. Yeah.
So do we have any questions for Jens, Max, or myself? This is a last opportunity. Yeah, let's see.
Hi, and thank you for a very interesting talk. I'm just thinking there seems to be a correlation between lifestyle and all of these diseases, and also what we eat, and that there are negative and positive effects by diet. And maybe you could comment on that. What should we eat when we leave here?
Yeah, I think Fredrik maybe can answer it later on the microbial side, but I think it will be based on again how your body digests the food and how those little compounds, those metabolites, affect your genes. Because I've heard people that say, I feel so well since I've stopped eating meat, so they became vegan. And if we study those metabolites, those little compounds in the blood, they're different than me, who I'm an omnivorian, for example. But the thing is, so there is no, it will be a personalized kind of advice. What is good for you will depend on your genes, your bacteria, to which kind of medication you've been exposed a long time. But I guess we will get there earlier than the treatment with these novel bacteria. We will get to the part where the doctor, who does not usually know that much about diet, takes your stool, takes a questionnaire of the diet, puts it together in an app or an algorithm, and those apps are already there in Israel. There are companies that do this, and it comes up with a list of food that if you eat that you have high glucose and you're prone to get diabetes, and a list of food that helps you to keep the glucose low. But it's not that eat fibers keeps you well or eat yogurt. It will be. I saw people that had lower glucose levels when they eat pizza. They are right. So this is what it is. It's really. It's all how the system interconnects, as Jens said, in your gut. So I think this will be the first, and I think it will be in a few years, because in Israel you can already take your stool, send it to the company, and they will come back with these dietary advices.
And the interesting thing in that study is that the computer was better than the dietitians. Yeah. So they actually did a controlled study, an intervention study, where they had half of the subjects went to the dietitian and half went to their algorithm, and the algorithm outdid
was better.
The nutritionist.
But it was a small study. It was 30 people. But need to say that, but it's.
But what it comes back to was that it's sort of based. If you look at the GI indexes that are so popular here, they're basically made out of the average of 10 individuals for each of these compounds. And what you see is that some people, as Max said, will have no glucose effect at all as the pizza. Yeah. Someone else has a huge effect, and other people would have an effect on broccoli, whereas others will not have.
Yeah.
So it's coming back to this individualization or personalization of dietary advices.
Yeah. But it will be a revolution for doctors, because we're pretending to know a lot about food, but we don't. And we don't. The whole first world kind of hospital is not fit for this. We don't have enough dietitians. We usually say, no, we check the box, eat less or eat less and eat better, and then we send the patient out. But now if you can have an app which reminds you every day, 3 times a day, what to eat, this might help, right? So I'm quite enthusiastic about exactly your question, that will help humankind.
Question in the back there.
Have you changed your own diet Yes. No, there's two things I changed. One is that I practice what I preach for my children. So when my children were born, we were living in the US, and you had to wash your hands all the time. We stopped with that. So we have a very unclean household. And the other thing is, except for one, the other two children had never had antibiotics, and the one was sick, so he needed it. So that's what I preach. For my diet, I started to eat less meat. That's the only thing I can say. So, because I checked my own microbiota, I have a very large family history of type 2 diabetes. Everybody of my uncles and aunts have it. I know I have a predilection to it. And I know that I'm not so good in digesting meat. But I like to eat meat, so I take it down. I'm like a flexitarian, where I go only 3-4 times a day, per week. That was a joke. Yeah, Freudian speaking. Yeah, I'm already thinking about the meal I will have tonight, and this will be huge platters of meat. Yeah.
My question was. You couldn't be guided by your own taste. It might mislead you. The last you said perhaps was the answer to my question. Should you listen to what you like and go for that?
Yeah. But the question is basically also, I can turn it around and say, do you know that we got microbes in our mouth that probably that are the first encounter of food that we have? So it might very well be that these microbes are on the taste bud and drive our palatability, so our appetite. So I think what we need to do with this field is really dissect, is it big? So the answer is no. I think the effect of microbes are important, but it's not a panacea. It's not going to explain everything. But the thing is, how big is it? And what is the hierarchy? So how important are microbes here? How important are microbes in the stomach for driving cancer or whatever? How important are the microbes on the skin and in the gut, and how do they talk to each other? And that's where we need the models of Jens. How do they talk to each other, and how can this one influence the other? And that, I think, will be very exciting.
Maybe I could give a comment about eating what you like. Yeah. I mean, there have been several studies that show that sugar is more addictive than cocaine.
Yeah.
And so, yes, it's maybe a good indication, but one has to be careful because many people also like to smoke, but we know that's bad. So,
yeah, that's why I showed the fructose slide. It's fascinating, and I know that there's a big lobby also for if you put a lot of fructose in the food, especially children, but also people try to take that food all the time. It's proven. So that's why the soda drinks are banned, at least in Europe, from cafeterias in schools, secondary schools.
I think we close the session here, and thank you for coming. Stefan wants to say something more.
Okay, I think first of all, I could congratulate us all being in the audience. I think this was absolutely fascinating. 3 great talks, giving different aspects, different angles, using different technologies of something that is extremely close to us all, food and gut microbiota. And we talked a little bit before, and I realize now how clear it is that it will be extremely interesting and have the same seminar in 2 years from now, because this is moving really fast with technologies like Jens is using, for example, and us all. So, and also I want to thank you all for giving such excellent talks with a small token of appreciation from Eva. Thank you, Max.
Thank you.
And Jens. And I have to disappoint you, it will not be meat for the meal. I saw it was some jello soup. But we should end with a big hand to you all. Thank you. Thank you.